All right, welcome everyone, both in person and online. We have with us today Dr. Kristina Mojica. She did her undergraduate degree in Hawaii Pacific University in marine biology, then moved down the road a little bit to the University of Hawaii, where she got her master's in biological oceanography. She did her PhD across the pond at University of Amsterdam in natural sciences, mathematics, and informatics. Excuse me. And then came back stateside for her postdoc at Oregon State University. I met Kristina last year when we were at sea in the Gulf of Mexico with Jeff Krause as well. And, we bonded over the amount of cruises and sea time that she's had. And that's what we do. We share stories about going to sea. And she's spent a significant amount of time doing open ocean microbial and viral ecology and oceanography as well. And she's going to be talking to us today about the ecological drivers of microbial interactions in the North Atlantic Ocean.
Yeah, thank you everybody for coming. I'm super excited to be here. So as Brandy kind of already spilled the beans, although my talk is entitled The Quest to Understand the Intricate Puzzle of Spring Blooms, I want to go a little bit beyond that and talk more generally about ecological drivers of microbial interactions. Okay, so start off with a little outline of my talk. I'll start off with some introduction on the importance of ocean and global carbon cycle. I'm sure you're all aware. The role of marine microbes, particularly the role of viruses, and where my lab and our research in the North Atlantic fits into this picture. I will then go into the main topic of this presentation, where I first talk about phytoplankton dynamics during the summer and spring in the North Atlantic, and then move on to cover some research about heterotrophic prokaryotes. Finally, I'll end with some final thoughts on the implications of all this, particularly with respect to the carbon flow through marine ecosystems. So this diagram illustrates the importance of the ocean and the global carbon cycle. The boxes are essentially stocks with their numbers there. The arrows are the fluxes in between those boxes. And the gold values is how those stocks and fluxes have changed due to the introduction of anthropogenic carbon. And so what you'll see is that the ocean not only represents a major reservoir for global carbon, but more importantly, it represents a major sink for anthropogenic carbon, storing close to 40% of the anthropogenic carbon produced since the beginning of the industrial era. Finally, the net uptake of carbon from the atmosphere, or net primary production occurring in the ocean, rivals that of land here, with the ocean accounting for 48% of the total production, despite only comprising about 3% or 0.5% of the total biomass. So biological processes in the ocean are what really regulate this carbon in the ocean, and it's through the balance between primary production by phytoplankton and bacterial respiration. So on the left, you have the classical food web where phytoplankton fix inorganic carbon and incorporate that into biomass, and they can be eaten by grazers, and then that carbon moves up to higher trophic levels, or also sinks out of the euphotic zone. On the right over here, we have our microbial food web, where bacteria then scavenge that dissolved organic carbon that's essentially lost this dissolved phase, reincorporate that into bacterial biomass that can be reincorporated into the food web or respired as carbon dioxide. So it's really competition between these three main fluxes: particulate flux, flux to higher trophic levels, and this remineralization that affect the carbon cycle and productivity. However, this picture is far from incomplete because it's not taking into it the role of viruses, which play a major role in marine biogeochemical and carbon cycles. So what do viruses do? Well, viruses essentially through lytic infection, infect their host. Here's your phytoplankton here, here's your bacteria here. They incorporate themselves in the genome and use that cell to reproduce more viruses. The end result is that they lyse those hosts to release out the viral progeny. The result is that all this intracellular material, which is rich in organic carbon and nutrients, is released into the surrounding water. So in this way, they essentially shunt that carbon away from higher trophic levels into this dissolved organic matter pool. Direct rates on estimating carbon flow through this viral pathway are rare, and consequently, we know very little about how they influence, how this pathway influences and how it responds to environmental conditions. This gap limits our understanding of biogeochemical fluxes over broad scales. So one of the main focuses of my research is to quantify these fluxes and what regulates the balance between microbial growth, essentially what's coming into this box, and loss processes, essentially what's coming out of this box here. With special focuses on how this imbalance influences population dynamics and biogeochemical cycles. In this talk, I'll present new insights on bacteria and phytoplankton mortality. Essentially, I'll be measuring these fluxes that are shown here in the stars for phytoplankton first. But I'll also go into the bacteria fluxes here. So again, these stars are what I go out and I measure. So this brings me to my lab, which we call MEL. Essentially, we are interested in how physical chemical variability, such as these mixing dynamics here or anthropogenic pollution, affect microbial interactions. So how microbes interact with each other, as well as how this interactions influence the flow of carbon. So how does this all fit together to affect the flow of carbon through marine ecosystems? Some people might have a pet project or a lab pet, but I have a pet ocean. And so this map is in the North Atlantic, which is my pet ocean. And it shows all of the locations that I've collected data since 2009. So over 14 years, with 168 stations within the North Atlantic Ocean. And I'll be presenting some of this data today. So you might ask, why the North Atlantic? Well, if you don't know. This is a GIF I put together, and it's created from 10-year monthly average MODIS satellite images of chlorophyll. And what you may notice is that the North Atlantic is a particular hotspot for changes in phytoplankton biomass, with much of this production occurring during the spring. This annual cycle plays a major role in ocean biogeochemistry and ecosystem ecology, and accordingly, phytoplankton bloom dynamics have been studied for over a century. Actually, the North Atlantic, remember when I said that the entire ocean takes up 48%, makes up 48% of the net primary production, the North Atlantic alone accounts for 20% of that. So this figure is just a different look at that same pattern. So what we're looking at the annual patterns in phytoplankton biomass per unit volume, which are shown in black here in the North Atlantic. And on the top here, we have changes in mixed layer depth. And on the bottom in red is the associated changes in photosynthetically available radiation, or PAR. And what you see is that the annual cycle on biomass shows a clear dependence on abiotic factors influencing photosynthesis and division, i.e. light and nutrients. However, even during the early days of Hawk and Grand, which was around 1912, it was recognized that loss plays a crucial role in the annual cycle of biomass, as changes in biomass are consequences of both changes in division and changes in loss. And only you can have an imbalance in these then you get a accumulation in phytoplankton biomass. Over time, however, historical studies found that grazing rates by zooplankton were too low to reconcile the observed accumulation rates in phytoplankton biomass. As a result, the bottom-up view took the forefront, and it became the predominant belief that factors regulating phytoplankton division rate determine where and when high concentrations of phytoplankton can occur, i.e. bloom. However, if we look a little bit closer at this traditional school of thought, what this really says is that accumulation rate should be directly proportional to division rate. So here you have accumulation rate, or R, or delta delta C. And so you should see a linear increase with accumulation with division. However, satellite observations have found that phytoplankton dynamics in the subarctic ocean reveal that phytoplankton division and accumulation rates are independent. This is because, again, accumulation rate is a product of both abiotic regulation of cell division and factors regulating phytoplankton loss. So factors regulating mortality are equally important to those regulating division rate. Indeed, when we look at phytoplankton loss and division rate, you can see that they're tightly coupled. What this means is that phytoplankton blooms are essentially a consequence of small imbalances between division and loss. So really just small deviations across this line. This has led to the development of the disturbance recovery hypothesis, which describes changes in the balance between division and loss over the seasonal cycle, which are driven by deep convection mixing processes. So we'll just go through this a little bit here. In this part, you have your phytoplankton decreasing, and over here you have your increasing. So this is actually when the bloom starts. You may notice that the bloom actually starts in the early winter, when phytoplankton growth rates are at their minimum and light is also at its minimum. The mixing rate is at its maximum. And so here in the summer, you have overgrazing and tons of loss and mortality going on because the water column has been stable for some while. So you have phytoplankton decreasing because your losses are greater than your growth. In the fall, autumn season, your mixed layer is deepening, and so your decreases in growth and loss are occurring simultaneously. Because you're diluting out your contact rate between predator and prey. Although you still have losses kind of decreasing, they're decreasing equally. In the winter, you actually have it where losses actually have a larger decrease than growth. At this point is when you actually have your bloom initiating in the winter. Once the mixed layer stops deepening, I was supposed to be doing this. Once the mixed layer stops deepening, phytoplankton and grazers concentrations rise in parallel, but there's ecosystem feedbacks because it takes a longer generation time for your predators to catch up to your prey. And you also have at the same time light-driven increases in division rate. And this maintains this growth loss imbalance and allows the bloom to kind of spring stratification period. This is just some additional evidence for this. And what is at the heart of this disturbance recovery hypothesis is that accumulation rate is proportional to the change in growth rate. In other words, division and loss are tightly coupled, such that accumulation rate, or the rate and change of biomass, is determined by the rate of change in division, which is driven by this temporary lag between growth and loss rate. So on the figure on the top here, the figure on the top panel shows the relationship between accumulation and delta mu using remote sensing data from the North Polar Zone. And what you can see is that the red line, the change in growth rate, is positive when you have a positive accumulation rate. These relationships are the focus of the next part of my talk, when I want to use field data to kind of back up what we're seeing from satellite. I first want to show that there is a tight coupling of loss and growth of phytoplankton, particularly in the summer stratified time period. And two, that this relationship between accumulation rate is really tied to this rate of change in division. So that's what I'll talk about here. I'm going to start off by talking about ecological drivers of phytoplankton annual cycles. So you may have noticed that I said that traditional grazing rates have not been able to reconcile our net accumulation rates that we have been observing. And that's because for a long time, two key players were not really taken into consideration. That's viruses and microzooplankton grazers. It is now recognized that microzooplankton grazing and viral lysis are an important source of mortality for phytoplankton communities. And these two have substantially different impacts on the flow of carbon through the marine ecosystem, as I showed with that wire diagram in the beginning. And one of the biggest problems is that they are seldom explicitly and completely parameterized in ecological and predictive models. Typically, ecosystem models only include a linear or density-dependent mortality. And what I hope to convince you of today is that the data that I will present highlights that such a general loss term are insufficient to capture processes underlying accumulations in phytoplankton biomass and the flow of matter and energy in marine ecosystems. So I just want to start off by kind of introducing the method that I use in the field. It's called the modified dilution assay, or MDA. The MDA provides simultaneous rates of phytoplankton division, viral lysis, and microzooplankton grazing. Essentially, you have two parallel dilution series. One is 0.45 series, so you just filter water through a 0.45 filter. This removes all your grazers, right? So grazers still have viruses present. You then have a second parallel series, that's the 30 kilodalton series that you run through a 30 kilodalton filter, and this removes both grazing and viral lysis. Essentially, what we're doing is we're diluting out the mortality pressure, and as a consequence, we see a resulting increase in growth. That resulting increase in growth is therefore the grazing or total loss rate. And the difference between those two lines is in my viral lysis rate. And we can literally, we do this for one liter bottles. We incubate them for 24 hours, and we can manipulate. I'll show you a little bit their light to do certain experiments with, but typically they are incubated under in situ conditions. One of the caveats is that mortality is assumed to be due to the encounter between prey and host and predator and virus. Although this might not always be the case, because sometimes you'll already have some phytoplankton that are infected when you start your experiment, but it's one of the assumptions of the method. So first I want to start off with data from two cruises in the North Atlantic, where the MDA was applied along a meridional transit in the North Atlantic, and then later within the gyre, Atlantic subtropical gyre. The map on the right over here, those white stations are all the stations where this experiment was conducted. And during these cruises, it was highly stratified, so strongly stratified according to depth average, 100-meter depth average, buoyancy frequency being greater than 5 times 10 to the negative fifth per second squared. The ODV plots over here show the corresponding vertical gradients in density on the left and phosphate on the right over the latitudinal transect, just showing the strong density gradient and subsequent nutrient limitation in the surface waters. You have your mixed layer depths being shallow, and those are indicated by these yellow circles, and also that also in the red is where I actually did my sample. So even though you have this strong horizontal gradient in density and nutrients, there was a slight relaxation here in this northern region, and that will come into play later in my results. And so what we found was that total loss, that is viral lysis plus microzooplankton grazing, was highly correlated to division rate for all phytoplankton groups measured under these strongly stratified conditions. These results indicate that there is a high turnover of biomass. More importantly, these results show that these two loss processes alone were sufficient to balance phytoplankton division rates and reconcile the lack of accumulation in phytoplankton biomass under strongly stratified and oligotrophic conditions. Now, if we take this total loss and we separate it out into the virus and grazing components, the relationship looks like this. So what presented here on the x-axis is the different phytoplankton groups or host groups with their loss rates separated into grazing, blue bars, and viral lysis, red bars. And I want to remind you that before any of these kinds of experiments, it was assumed that 80% of phytoplankton mortality was due to grazing. What this data shows is that over these broad scales, viral lysis and microzooplankton are comparable for all phytoplankton groups measured. So this is really important information into how ecosystems function. Remember, viruses are really recycling that carbon's not moving up the food chain. So if you think about the blue bars representing the classical food web, that is the energy transferring up the food to higher trophic levels, and the red bars are recycling. This recycling of organic matter is important because it enhances bacterial microbial mediated remineralization, and therefore it has important consequences for how carbon flows to the ecosystem. What's more is if we look at how these loss processes are distributed over our latitudinal gradient, they are not random. There is a pattern to it. So what I'm showing here is the log of the ratio between viral lysis to grazing rate over the latitudinal gradient. And the dotted line shows where those two processes are equal or in balance. So everything above that line is viral lysis dominates, and everything below that line, grazing dominates. What we found was that really within our highly stratified oligotrophic region, this is KT turbulence here, viral lysis was the dominant mortality factor. And you saw this striking switch in the northern latitudes where we had this kind of relaxation of strong stratification, where it really switched over to grazing dominated. And this is one of the first studies to show this, and I'll kind of show you that this is kind of consistently found now. So this was like really exciting, but also kind of scary. And what we found was that this trend was driven by a loss of viral lysis rates, which was linked to this eddy diffusivity. So really, which is a measure of turbulence. And this suggests that viral activity is negatively impacted by mixing processes. Again, the first time we found this, but I'm finding it over and over and over again. So now I want to go into results of more recent cruise in the North Atlantic that took place in the spring of 2018. Again, a series of modified dilution experiments were performed along a diagonal transect this time here. And in the first section of the cruise, you can see that it is, they were classified as weakly stratified. And the northern section shown in the red box here were classified as non-stratified, again, according to this 100-meter average buoyancy frequency, being less than 2 × 10⁻⁵ per second squared. And from the density plot, you can notice that there is very little vertical gradient in density in most of these stations. So on the right, I'm showing a similar plot as before, comparing total loss, now on the x-axis, which again is viral lysis plus microzooplankton grazing for the different phytoplankton groups, versus division rate on the y-axis. And now you'll see a clear difference from what we saw in the summer. where spring actually has division in excess of loss. That's kind of what we expect in the spring, right? We're having a positive accumulation rate. And what's shown on the right in the hatch bars is that over the entire transect, this resulted in positive accumulation rates for the different phytoplankton groups. So again, when I take that loss and divide it out into its different mortality components, we see again a different picture in the spring compared to summer, with microzooplankton grazing having much higher rates compared to viral lysis. This time, we found a significant decrease in the virus grazing ratio with latitude consistent with the decrease in stratification index. This time, we didn't measure microturbulence, so we had to relate it to buoyancy frequency. But kind of the same indication, right? That these viruses are being negatively impacted by turbulence. And this corresponds again well with what we saw in summer. We saw that switch to grazing dominated in that northern region. Also during this spring cruise, we examined the implications of altering the light environment experienced by taking phytoplankton population from a deeply mixed surface layer and incubating them under static simulated shallow conditions. In other words, we compared the impact of light on the balance between division and loss by taking a phytoplankton community acclimated to mid-mixed layer light conditions, which are shown in the black symbols here, versus those incubated under simulated light climate of the 15-meter water depth. Those are shown in gray here. The resulting plot then shows the comparison of PAR under incubation and in situ conditions. On the left over here, we see the response in phytoplankton division rate versus delta PAR, which is the difference between incubated and in situ PAR. And what you see is that these changes in division rate really remind you of a typical photoirradiance curve, which is actually indicated by the solid line where I added that model here. So another important part is that when we did this, we actually found no significant effect on mortality rate because this was all within that 24-hour period. So remember I told you that the positive accumulation rate has to be due to the lag in the response of mortality to actually respond to those changes in division. So we already kind of see that here. So in other words, phytoplankton growth was accelerated by increase in abiotic light conditions and decoupled from mortality. Again, due to the lag in the response of mortality over that 24-hour period to this increase in division. And this resulted in enhanced accumulation rate. And what's shown in this figure is the resulting rate of change in division versus accumulation rate, where accumulation rate is the net consequence of division minus loss. So this was the first clear experimental demonstration of the central tenet of the disturbance recovery hypothesis, specifically that phytoplankton accumulation rates are quantitatively linked to the rate of change in division. So where are we now? Well, my PhD student that joined, I think 6 months ago, is now looking at a cruise that we did in the height of COVID in spring of 2021, where we did similar perturbation experiments now down here. But this time we did both accelerations and decelerations in growth. And so what we want to see is kind of the opposite. Can we get it where we actually have a negative decrease in phytoplankton biomass? And so that will be coming soon. I am also working on another cruise that looks at a longitudinal transect, one of our first ever. And this is actually in the equatorial zone. So much different conditions, much more stable. We don't have this really strong dynamic and mixing. Because of this, we have found so far that the phytoplankton growth rates are low and mortality largely outweighs growth. And there's also an extra confounding factor is that this area is highly influenced by iron deposition from the Sahara dust. And that was actually one of the main aims of this cruise. So we do have dissolved iron and dust data on that. So if you're interested in that, I'll be at OSM and presenting a poster on that. So I've been really focusing over here on our phytoplankton for the first part of my talk. But remember, I said at the beginning of this talk, the ocean carbon cycle is regulated by the balance between phytoplankton and bacterial respiration. So now I want to really move to the right side of this diagram. So for the second part of my talk, I would like to talk about the ecological drivers of heterotrophic prokaryote populations in the North Atlantic. So again, I'm going back to that original cruise transect. We call it stratified. During the summer stratified conditions, here's again, just remind you of that density. But now I'm looking at bacteria, heterotrophic prokaryotes, and their mortality processes being bacteriophages and protists, particularly heterotrophic nanoflagellates. So in this figure, I'm showing the ratio of virus-mediated mortality on the top, these guys right here, VMM, and protist-mediated mortality over the longitudinal transect. And the gray area shows the stations that were classified as oligotrophic based on nutrient and chlorophyll concentrations. And what you see is that over much of the transect, viral-mediated mortality was the dominant mortality processes, contributing on average about 55% of the total mortality. And although you don't really see a clear pattern in this ratio over our latitudinal transect, we did do an RDA. I tried to spare you from looking at too many RDAs if you guys are not familiar with them. People hate them. I will have one later on for all the ecologists out there. But I wanted to limit it, but we did an RDA. And the interesting thing is that these two mortality processes were regulated by different physical processes. And so grazing was positively influenced by temperature, right? And viral lysis was again negatively influenced by vertical mixing, KT. And so they actually were increasing in the same direction and decreasing in the same direction, but again, regulated by different physical processes. What's more is that these two mortality processes can sometimes be negatively correlated. Here I'm looking at viral abundance versus heterotrophic nanoflagellate abundance. And you see that in the shallow layers, they were positively correlated. In the deeper layers, they were negatively correlated. This was the same for the production rate. They are also tied in a similar way. And this was really kind of linked to the abundance, where the abundance of their host or prey organism was significantly less in the deeper layers. And so you can think about this as if there's plenty of prey, they're both kind of eating them, lysing them, and so they're both increasing in parallel. However, when their prey becomes limited, they start to have an inhibitory effect, right? And also a kind of confounding part there is that heterotrophic nanoflagellates can actually eat viruses, although it's not shown to be like really significant, although there's only a few studies. And they can also eat virally infected prey. So they could actually reduce the production rate because they're eating hosts that are already previously infected. Yeah, so they could compete for the same resource. So you might have some exploitative competition going on when hosts are low. Finally, when we look at total microbial mediated mortality, which is TMM here, versus total available carbon, which is the standard stout carbon plus production, what we see is that over the latitudinal gradients, heterotrophic prokaryotic community moves from a near steady state system over here to a system regulated by high turnover to net heterotrophic production in the temperate region. So everything below here is net heterotrophic production. I'll come back to what all this means in a few. So another thing to consider when working in the North Atlantic is that this ocean basin has an energetic eddy field. So if you look up on the right here, what this is the animation that shows dynamic eddy field in this region based on sea level anomalies, where your anticyclones are shown in red and your cyclonic eddies are shown in blue. And these areas change the sea surface because they have the ability to interact with the isopycnals, either doming or depressing isopycnals, and getting a response in your phytoplankton community as a consequence of how they interact with the nutricline. So here we can see these effects here on your chlorophyll concentration, either a negative effect or a positive effect. So we know quite a bit of how phytoplankton kind of interact with eddies and how they influence their production. But what we don't know is how these influence the bacteria community within them. So I was involved in 2 cruises in the Western North Atlantic. And these are the stations that I'm going to talk about, shown in this map. And these took place during the winter of 2015 and the spring of 2016. And so to get a different dynamic range of physics, we had 3 basic categories. That's the anticyclones shown in pink, your cyclonic eddies shown in blue, and then we had some stations outside eddies. And so one of the things that I was interested in is these populations that we can distinguish on the flow cytometer. They are called low nucleic acid and high nucleic acid populations. We call them that because in bacteria, your green fluorescence is directly proportional to your nucleic acid material or your nucleus. And so another thing, there's been some kind of conversation from sorting out these populations that there's some certain signatures of bacteria that comprise these 2 groups, wherein HNAs are copiotrophic members and LNAs are leukotropic members, such as SAR 11, for example. And so I was interested in whether we could link these with the physics and what does this mean for the virus community. So one of the first results I found was that the ratio of HNA to LNA cells in the community is related to the overall abundance of heterotrophic prokaryotes. Moreover, what we find is that the ratio is predictive of heterotrophic prokaryote specific growth rate. In other words, the specific carbon uptake of the cells, suggesting that HNA cells take up more carbon than LNA cells. So what is driving the distribution of these cells in the community and therefore the total heterotrophic production? Well, I ran an RNA analysis. Again, I'm not going to make you look at those, but it revealed that phytoplankton carbon, DOC, and eddy field explain 84% of the variability in the abundance, composition, and productivity of these 2 groups. And that the heterotrophic prokaryote abundance, specific growth rate, and fraction of HNA cells were higher in cyclonic eddies, consistent with higher phytoplankton carbon. So I just want to sum up what I just told you is that the eddy field is really driving the availability of phytoplankton carbon and DOC in the marine system, which affects the abundance, composition, and production of my bacterial community. So the question is, how does this influence my viral community? So here's that RDA I was promising. So really what I wanted to know is are these groups important for the dynamics of my viruses? So let me walk you through this. First, if you have a keen eye and you know RDAs, you might have noticed that this figure is actually showing you not the 2 typical first axes. which are RDA 1 and 2. I'm actually looking at the second and third axes. And that was because I was particularly interested in this one right here, which was most represented by this third axis. So I just manipulated that multi-dimensional space there to look at that a little bit better. So what this really shows is if you have your explanatory variables here in black and you have your response variables in red, the length of those lines is how much those are contributing to your model. And if they're closely, if the lines are close together, that means that they're significantly correlated. So again, I already kind of told you that specific growth rate, heterotrophic abundance, and the ratio of HNA to LNA are all related to each other, all correlated to each other. And what you see here is that we have these different virus groups, V1, V2, V3. And now you can see that V1 and the production of V1 was most closely tied to HNA. And on the opposite, which would be LNA, you have your V2 abundance and production. And this is your ratio of your host to viruses. So what this tells me is that these populations, or the relative distribution of these two different groups, has some influence on the distribution of my virus group. This is interesting because now this is saying that these flow cytometry groups have some kind of ecological implication. And so if I look at this a little bit closer, now looking at these V1 viruses versus HNAs, you can see that they're highly correlated and LNAs with V2s. What's more important is that HNAs are the most productive cells, as I already suggested. And so if you think about the killing the winner hypothesis, that viruses are going to infect the most active and abundant hosts out there. You see that's true for V1 is really going after HNAs and V2 and LNAs. And so these suggest that there's some group and activity-specific linkages between these prokaryote hosts and virus groups that we can distinguish on the flow cytometer. And what's more is that depending on who dominates the system, it has implications for how carbon's moving through my ecosystem, because the V1 HNA group has 2 times higher production rate compared to your V2 populations. And again, HNAs are the most productive hosts. So what does this mean? So if we look at what this means, whether we're in a cyclonic eddy, you would have consistently higher phytoplankton carbon. Your HNA V1 group would most likely dominate, leading to a higher recycling of DOM, enhanced respiration of organic carbon due to microbial loop, which can reduce vertical or horizontal export potential of DOC from the local environment and reducing CO2 drawdown. Because remember, recycling of DOM ultimately enhances organic matter remineralization and therefore reduces the potential of transfer up the food web via bacteria. So where are we now? This is another PhD student, Ian, and he is working on a spring cruise to see if we could still find these kind of relationships in a much more dynamic mixing condition. So that, because I didn't look at that in the summer cruise. So he's looking at this and he has already found the same similar links between HNA and V1 populations. And he's going to discuss that in terms of carbon flux over the seasonal stratification cycle. And so again, he's going to be presenting this at OSM. So check him out. So finally, you might be asking like, oh, God, that's a lot of rates you're measuring. You're talking a lot about this stuff. What does it all mean? Well, I would like to kind of sum it all up with some final thoughts on why these are important with respect to the carbon flow through marine ecosystems, and particularly that we are not currently including these in ecosystem models. So this is a very seminal paper from 1999. in which Curtis Tuttle and Stephen Wilhelm, really well known in the viral ecology field, they produced this kind of, they call it the back of the envelope calculations on how much carbon flows through the viral shunt. Again, this carbon flow, this is in terms of photosynthetically fixed carbon. And they found that as much as 1/4 of the organic carbon flows through the viral shunt, 6 to 26%. Nobody has since that time put numbers, actual numbers and rates. They used like a handful of rates that we actually had back there in 1999. And everybody still quotes these right here. That's 25%, very significant. Well, if I take that and I put what I have, where I have the same rate measured on the same water, which I only have for one cruise during the summer, and we look at that under summer stratification, and we see that instead of 80% being going to grazers, remember I told you that was the traditional school of thought, right? We only have 24 to 25% going to grazers and 21 to 35% going to the viruses. And over here for the bacteria, we have 3 to 9% going to grazers. That was 36%, because back then we didn't know the importance of bacteriivores too much. And then 3 to 15% going to the viruses, where I have 12 to 48%. So instead of 6 to 26%, I have 33 to 80% of the photosynthetically fixed carbon moving through that viral shunt. It's extremely significant. Where am I now? Well, I'm waiting on getting more funding to actually go out and measure this during the spring and over more stations where I actually match rates, right? Because I believe that you need to match these on the same water source, do all these experiments on the exact same water. So I'm working on that. So kind of just to sum up everything, I can sum them up pretty much in 3 points here. The data I presented shows the importance of virolysis and microzooplankton to regulating phytoplankton communities and heterotrophic prokaryote communities in the North Atlantic. Physical disturbance can alter the balance between division and loss, where virolysis seems to be negatively affected by vertical mixing processes. And the partitioning between microbial biomass and these mortality modes as a consequence is affected by vertical mixing. And more importantly, the data that I presented today demonstrates the importance of incorporating microzooplankton grazing and virolysis in numerical and biogeochemical models in order to fully understand the balance between energy and matter and marine ecosystems. So with that, I have lots of people to thank, because again, there's lots of cruises, lots of people. We have our ocean there and our viruses, because without them being so cool, I would never be so cool. And of course, my supervisors along the way and collaborators, they are amazing. So with that, I would like to thank you guys and welcome any questions.